I still have a few people who haven't turned in your syllabus form, and so if you haven't done that syllabus form, if you would sign that and get that to me, I would appreciate it. Okay, I have a few questions about the mastery quiz. If you do need to retake the mastery quiz, there will be a new quiz. It won't be just the exact same quiz you took last time. But similar types of questions and concepts, and it's not unheard of for a question to actually migrate over from the first quiz to the second quiz. So make sure you sit down and look at each of the answers on the quiz that you took, whether you got them right or not, to make sure you understand why that is the correct answer. Because sometimes you're guessing and you just happen to luck into the correct answer. So please make sure you understand the concept that's being tested. You can use a page of notes again, and if you want to revise those notes, that's fine. If you want to just add to them or something, you're able to pick up your old quizzes. You either got them in lab or they're in Hadley's office. The intent is to get you to go over the material and be comfortable with it. And then in terms of taking it, I will give some copies to Dr. Morden, and so if you want to take a retake at the end of your lab section, that would work. If you want to drop by my office sometime when you're free, you can come by and I'll give you a copy and you can just go in the lab and take it. Or if that doesn't work, we'll have some time at the end of class on Friday where you can do a retake. And hopefully we'll be done with it after Friday. Okay? So any questions about the master's test? Any questions about anything else? I will have to check for you. If you want to take advantage of the recordings that we're doing of the class, they are not on the cell biology website. I think they're on the Natural Science Division website page. I'll check with Natalie to make sure and send you a link for that. But I think she had difficulty getting it onto the cell biology website. So they'll be posted in a little bit different location, but they're still pretty easy to find. Okay. Sound appealing? How many of you would like to be able to fly? Or breathe underwater? Or swimmers. Most definitely. This is not an unheard of belief, particularly in Native American cultures. In Native American Indian cultures, it was widely believed that if you ate the liver of a bear, you would gain tremendous strength. But in reality, can any of these things happen? Meet the emerald sea slug. Is this a plant or an animal? What about regular slugs? Plant or animal? Animals. Okay. What about this guy? What about the emerald sea slug? This is a marine slug. What do you think? Plant or animal? What might cause to give you pause a little bit? Might just mean say it's a slug, it's an animal. What about that image might cause you to question a little bit?
It kind of looks like a leaf, like it could photosynthesis.
Looks kind of like a leaf, okay. It's got a lot of surface area. What's even more kind of jumps out at you? Pardon me? It can live underwater, okay. Is that necessarily a plant attribute, though? Be able to live underwater? No, not necessarily. What should just really jump out at you looking at that? What screams out potentially plant?
The veins look like they're part of the plant.
Okay, it does look like that there are veins in here that look like they're the veins in the leaf. You're making it too hard, folks.
It's green.
It's green. Okay. What makes things green?
Chloroplasts.
Chloroplasts and a pigment inside called chlorophyll. Okay. And indeed, if you cut open the tissues of this particular sea slug, there are chloroplasts inside. Where are you supposed to come from? This is an animal.
Does it come from the algae that it eats?
It comes from the algae that it eats. Okay? Eat an algae, photosynthesize. The emerald sea slug can actually do that. Now, it's not uncommon for organisms to establish a relationship where one organism lives inside of another one. You know what you call that phenomenon? Two things are kind of coexisting together and benefiting one another. Symbiosis, okay. And actually there's a symbiotic relationship between an organism that you used in lab last semester and a protozoan, and that is the termite. There are protozoans that live in the gut of a termite, and those protozoans produce an enzyme that can break down cellulose, and so termites can eat wood. Okay? The termite cells themselves do not have the ability to produce that enzyme, but the protozoan does. Yes.
Don't flamingos, like, since they eat shrimp, it changes the...
Changes their pigmentation. Yes, it does. Absolutely. But what's really interesting about the sea slug is it's not just that there are little algae living within its tissues. They actually break down the algae and they incorporate the chloroplasts of the algae into their cells. That's very cool. But then they do something else that's just, for molecular biologists, kind of mind-blowing. They can not only steal organelles from another cell, but they can steal genes from another cell. And they can actually incorporate algal genes into their genetic material. That's called horizontal gene transfer. And this is the first demonstrated example of horizontal gene transfer occurring between multicellular organisms. We're going to do some horizontal gene transfer in lab later in the semester. And you're going to transfer a jellyfish gene into a bacteria. The bacteria are multicellular. So in the emerald sea slug, they can feed on algae, steal their chloroplasts, steal some of their genes, and this allows them to temporarily live like a plant. They can photosynthesize. And they can even pass some of this ability on to their offspring, which is very, very unique. So why does it help you get a gene? Well, they take some of these algae genes and they stick them into their chromosome. That's why it can potentially be passed on to offspring. Because every time the chromosome duplicates, those genes are duplicated as well. And these genes help to repair damage to the chloroplasts and keep them functioning. So this sea slug doesn't need to continually keep consuming algae. Once they've got the algae, the chloroplasts, and their genes, they can retain this power of photosynthesis for up to 9 months. Very, very interesting things. I'm going to try as much as I can sometime during a class period to throw something out that's kind of current science news that relates to things we are talking about or will be talking about. And we certainly will be talking about later this week basic cell structure, and so we'll talk about the function of chloroplasts and mitochondria and all that stuff. But additionally, we will also talk about photosynthesis in a lot of detail later on. So keep your eye out in the news for the emerald sea slug. It's pretty hot stuff right now. Okay, so I showed you a couple of pictures last time. And you drew a couple of conclusions from looking at those pictures that the images suggested that all living things that we know of to date are composed of one or more cells, and that all cells come from other cells. And those are the two basic tenets of the cell theory. Remember at the end of the period when I said about the cell theory was formulated? Civil War time, around the 1860s. It took a lot of time, though, for the cell theory to develop, in part because cells are really small. And because cells are really small, we need certain technologies to be able to observe them, to even know that they exist, and then finally to dig into them and find out more information about how they function. So here's a little scale right here that shows you various sizes. And when we talk about looking at cells, we're working here in this micrometer size range. Okay? And if you look at this side over here, you can see that things that are this small cannot be seen by the unaided eye. We need some kind of optical instrument to be able to extend the power of our eye and to see things that are smaller. And so these are visible with a light microscope. And then additional technologies, the electron microscope, allow us to see things that are smaller than cells, cell parts, for example. So one of the things that we will be doing in lab next week is you'll be doing a microscopy lab where you're going to get comfortable with use of the microscope and to look at some of these cells and find out some kind of representative cells and look at them kind of from an evolutionary standpoint. Look at prokaryotic cells, eukaryotic cells, single-celled organisms, multicellular organisms. So the study of cell biology is really very closely intertwined with the history of the development of the microscope. So I want to spend just a couple of minutes talking about microscopic technology, microscope technology, and then we'll show you some advances in technology that have taken place since the first microscopes were developed. If we go back to the mid-1600s, one of the people who's kind of credited with being kind of the father of microscopy is this Dutch guy that's shown over here, Anton van Leeuwenhoek. And this is a sample of his first primitive early microscope. It's basically almost a little handheld lens. Van Leeuwenhoek ground all of his own lenses. He was a very accomplished scientist, but he was also a Dutch linen merchant. And one of the reasons he developed his little handheld microscopes was to look at the cloth that he was buying for small flaws in cloth. But he was also a very curious person, and he started to look at things other than linen, including samples of pond water, for example. And in them, he was able to observe tiny microscopic creatures, some of which are shown here in his original illustrations. And he called these things animalcules. Tiny microscopic animals that no one had ever seen before. When he reported his findings to the Royal Society of London, which was one of the preeminent scientific organizations of the time, I mean, it was headed up by Isaac Newton at one point in time, to give you an idea of its stature. It just blew their minds. Because all of a sudden people realized that there were all these little tiny critters out there in water and food and the air that they had never been able to see before. Really scared a lot of people, but it opened up for the first time this realization that there was a microscopic world that we weren't aware of. Anybody know where the word cell comes from? Why do we call these things cells?
Somebody looks in the microscope at cork and it looks like little cells, like an actual cell.
Yep. Robert Hooke, who was also in the Royal Society in the mid-1600s, took very thin slices of cork and looked at them with one of these primitive microscopes, and he saw all of these little repeating structures that looked like prison cells. So we called them cells. If you know anything about cork, cork is a dead tissue. And so what are the boundaries of these little cells right here? What are they made of, do you know? Yeah, we're really looking at a cell wall, basically. All the contents of the cytoplasm have died. So a whole bunch of cellulose basically makes up these cork cell walls. Well, as people started looking at more and more things with better and better microscopes, they started to see that these cells were kind of were a universal component of all living organisms. Some organisms were only a single cell, but many organisms were multicellular. So as microscopes improved, our knowledge of cell biology improved. Now, there's a fair amount of information on this slide, and I'm not particularly interested that you memorize all of this information for an exam, but I think it's important to kind of see the different images you can get from the different types of technologies that we now have available to look at cells and tissues. These are all examples of light microscopy, and so here we're using light passing through lenses to observe various kinds of tissues and cells. Bright field is what we'll be using in our lab. And this shows you an unstained image and a stained image. And on the stained image, you can start to see a little bit more cellular structure. You can see a nucleus, for example. But we won't use this in lab, but we have one. There's also fluorescence microscopy, which can actually use fluorescent dyes to bind to certain portions of a cell and highlight those. And you can see some real detail you can't see in this kind of an image. There is phase contrast, Nomarski, confocal. All of these give you a little bit different image and a little bit different insight, and they're all kind of improvements in technology that have happened over time. This just gives you a little bit better, larger image of regular bright field and unstained cell, like you'll see next week, stained, phase, Nomarski, fluorescence, confocal. Start getting some pretty interesting pictures of cells. But light has limitations in terms of how much detail you can see when you view things using light as your energy source. One of the two keys that's really important for seeing things, obviously, is magnification. You've all used a magnifying lens at some point. This makes the image look bigger. But what's really also very important is the concept of resolution. Resolution has to do with detail. Does anybody know how resolution is defined? Resolution refers to the ability to see two distinct points as actually being separate. So if I've got two little dots right up here, these are separate dots, but at some point your eyes, when you get far enough away, will not be able to see those as individual entities. They'll blur together. And so if you have less resolution, you can see less detail. So one of the things that microscopes have to be able to do to allow you to see more about a cell is to not only make it bigger, but also increase the resolution, increase the detail. Because if something's blurry, just making it bigger just makes a bigger blur. So in the 1950s, a new technology came out that just opened up a whole new world of microscopy. And this was the development of the electron microscope. So now instead of using light to view specimens, use electrons. And if you're interested, the shorter the wavelength of this energy you use to view an image, the more detail you can get. And electrons have a shorter wavelength than visible light does. So that's one of the reasons you can get more clarity. There are two basic types of electron microscope. There are scanning electron microscopes and transmission electron microscopes. And scanning electron microscopes primarily look at surfaces. They can give you really, really incredibly detailed three-dimensional images. Transmission electron microscopes involve taking tissues and cells and making very thin sections of them, and then looking at those stained thin sections with the electron microscope. So here, you can see a lot of intracellular detail. You can start seeing cell organelles, for example. So, here is a scanning electron microscope image of a cell, a human cell, in your trachea that has these little tiny projections sticking out from the cell surface called cilia. Does anybody know what your trachea cilia are responsible for doing and how they help protect you? You know your throat is lined with cells that have all these little hairs that beat back and forth?
Doesn't it keep like dust and other particles that you don't want going into
Yep. Yep. Kind of sweep stuff back out of your respiratory tract and prevent stuff from infecting your lungs. Well, here's a scanning electron microscope image showing you a very magnified and high resolution image of these individual cilia. Here is the same tissue sectioned, this is my laser pointer, with transmission electron microscopy techniques to show you the ultrastructure of these by kind of slicing them in half. And so you can kind of see internal images. Okay, so scanning, transmission. Well, as a result of this, a picture has kind of emerged of what cells typically look like. And you'll oftentimes see in a textbook an image like this that shows you a generalized animal cell, for example. Now, based on what we saw the other day, what's wrong with this idea of a cell? Do all cells look like this? No. Did the muscle cell look like this? Did the neuron look like this? No. So one of the reasons I really don't like images like this that much is it implies this idea is that there's just kind of one type of cell. There's one type of animal cell, one type of plant cell. Another thing I don't like about this image is that it's two-dimensional, and the cell is a three-dimensional structure. And so it's really kind of hard to appreciate the dimensions that really exist by looking at this two-dimensional picture. And the thing that I really don't like about this kind of image is that this is a very static image, and it really implies that there's really nothing going on inside the cell. And that is really totally wrong. The cell is just a dynamo of activity. Some interesting projects have been done, most recently by Harvard University, to try to create detailed animations of what it might look like inside of a real living cell. And I'm going to show you a little clip of this. It takes a couple of minutes. Some of you probably have seen it before in high school science courses. It's called Inner Life of the Cell. And it concentrates on one of the cells we're going to kind of use as an example, the white blood cell. You all have white blood cells circulating around in your circulatory system. And those white blood cells are patrolling your body looking for infectious agents. So what if you get a splinter that has some bacteria on it? And it is now introduced into your skin tissue. If those white blood cells are going to attack the bacteria, they have to be able to migrate out of the circulatory system and into the surrounding tissue. And the circulatory system is a series of tubes. How do you get out of the little arteriole or capillary and into the surrounding tissues? And these cells actually have to go through a tremendous change in their shape. And they basically crawl their way out between the cellular junctions of those little endothelial cells that line the capillaries. So the video we're going to see illustrates some of the changes that are taking place in this white blood cell during this process of moving through the circulatory system, grabbing onto the cell wall or the cell membrane at some point in time of an endothelial cell, and then crawling out of the circulatory system into the surrounding tissue where they can attack bacteria. Much of this won't make sense to you now as you see it, but I want to show it to you for the first time, and then we'll come back after we do a tour of the cell and show it to you again, and a lot of the things will make a lot more sense. But one of the things it really does illustrate is how dynamic cellular processes are and how much stuff is actually going on inside of the cell. Let's take a look at this for a second. There's your white blood cell. So again, a lot of that probably, made no sense to you at all in terms of what was happening and what was going on. Some of the parts you may have recognized. Hopefully you'll have a little bit better insight in what some of that is by the end of our discussion of the tour of the cell. But obviously that represents a much different image of what's happening inside of the cell than this does. Okay? Another technology that's really important is a technology that allows you to study one part of the cell in isolation from the rest. Okay. So what if you were particularly interested in cell energetics and you wanted to understand what this guy does? What cell structure is this? That's a mitochondria, okay, sometimes called the powerhouse of the cell, responsible for cell respiration and making ATP. So if you wanted to study the mitochondria and you had a bunch of rat muscle tissue, for example, what's the first thing you'd have to do to that tissue to start getting those mitochondria to study? Where are those mitochondria located? They're inside of cells, right? And the cells are part of this connected muscle tissue. So realistically, if we want to get mitochondria to study, what are we going to have to do to that tissue? Bust it open, right? Okay. Somehow break it up and start separating the cells from one another. And then also breaking open the cells. And so all of the contents of the cell go rushing out. That particular technique is called homogenization. And there's all kinds of different ways that you can homogenize tissue. You all familiar with a mortar and a pestle? Those little white things and a little handle that you can grind tissue with. Okay, so you can put tissue in a mortar and pestle and grind it up to homogenize it. You can throw it in a blender and just blend the heck out of the tissue and break open the cells. So now we've got a liquid that contains parts of broken cells, some unbroken cells, all kinds of cell organelles. If you want to get rid of some of the junk that's in this liquid, some of the big stuff, you can do a simple filtration, like through filter paper or cheesecloth or something, and catch some of the big stuff. But then this liquid, this filtrate that you collect, still has a mixture of all of these cell organelles. And you want the mitochondria. You don't want the nuclei. You don't want the Golgi apparatus. You don't want the endoplasmic reticulum. You just want the mitochondria. So there's another technology we can use to start separating these individual cell organelles from one another. Do we know what that technique is?
Can you spin it?
We can spin it. We can spin it in a device called a centrifuge. And the faster you spin it, the more that this liquid is subjected to centrifugal force. And things that are in that liquid will begin to separate out and move toward the bottom of the centrifuge tube based upon differences in their density. So the bigger things will spin down faster than the smaller things do. So there's a technique we can use to isolate cell organelles called differential centrifugation. Here's how it works. Okay, so we homogenize tissue in some way. Okay, we start out with some tissue cells. We throw them in a blender basically. That'll start breaking up the connections between the cells, start breaking open cells. And we've now got this homogeneous. We then take one of these tubes and put it in the centrifuge. Next time you're in the lab, if you look in the far corner over there, we have a large refrigerated centrifuge, and it's got a little head on it that's attached to a shaft, and basically that turns the head around and around and around. And I think that our maximum is about, 20,000 RPMs for that particular. So it gets going really fast. It creates a great centrifugal force. and then things start moving down the tube based upon differences in their density. So, what you can do is you can do a series of sequential centrifugations to start separating out various organelle-enriched fractions. So if we start out right here with our original homogeneous and spin it at 100 times g, 100 times the force of gravity, for about 10 minutes, only the most dense things will start pelleting out at the bottom. So things like nuclei and cell debris, which are all big. So they congregate down at the bottom, and this liquid still has everything else that's smaller. This is the pellet, and the liquid is called the supernatant. Well, if we're interested in mitochondria, they're still going to be in the supernatant. So what do? We carefully decant off the supernatant, leaving the pellet behind, pour that into another tube, and spin it faster. Let's say 20,000 times g. And if we do that now, that's sufficient g-force to pellet the mitochondria, but the smaller cell organelles will still remain back here in this supernatant. So, if you want to study mitochondria, you can now decant the supernatant and then use this pellet, and that will contain very enriched populations of mitochondria. If you want something smaller than that, then you would decant that supernatant, throw away the pellet, and spin it at faster speed. And now you might start getting cell membranes or other cell structures. And then if you still want smaller things than that, you pour off that supernatant and spin it faster, and then you could get a pellet that was really enriched in ribosomes, for example, which are very tiny. So by these series of differential centrifugations and succeedingly spinning the supernatants at faster and faster speed, you can start to isolate various cell organelles from one another. How could you be sure that you had mitochondria in this pellet? Is seeing believing, usually? So could you take a sample of that pellet and look at it under electron microscopy, for example, and see, are there primarily mitochondria there and not other cell organelles? Mm-hmm. And actually, there's another technique that you can use also to analyze these particular pellets. It's not uncommon for the membranes of cell organelles to have specific enzymes associated with them, because the organelle has some particular function, and enzymes help it do that function. So you can do something called an enzyme assay, where you look at the pellet and say, is this particular enzyme that's known to be associated with mitochondria in that pellet or not? And so you can actually have a profile of mitochondria should have these enzymes, endoplasmic reticulum should have these enzymes, and you can analyze your pellets to see if they have the proper enzyme profile. This is what I did for 4 years when I did my PhD, and Dr. Morton did a lot of this too. I was looking at plant tissue. You were looking at frog kidney tubules. Okay. And I was trying to figure out where a particular process occurs called glycosylation, which means adding sugars to proteins, inside of the cell's membranes. And so I had to bust open plant cells and isolate the various cell organelles and profile them and see which organelles I had in my particular little test tubes, and then there was a test we could use to find out where this glycosylation pattern occurred. Okay? So this is not uncommon scientific research for being able to study cell organelles in much greater detail. So let's take a little look at cell structure. There are some basic requirements that must be fulfilled if something is going to be considered to be a cell. It has to have, at a minimum, cytoplasm, DNA, ribosomes, and a cell membrane. Some cells, this is pretty much all they have. Other cells are much more complicated than this. They have many additional cellular structures. Until the electron microscope was developed, we didn't realize that there were fundamentally two different types of cells. But once we saw images of a transmission electron microscope of a bacterial cell and a human kidney tubule cell, that there were some just really fundamental differences in the way they looked. And that then was established in this concept of prokaryotic cell type and eukaryotic cell type. Okay? What are some differences between prokaryotic cells and eukaryotic cells? Someone say something. A nucleus and other membrane-bound organelles. Which one happened? Prokaryotic or eukaryotic?
Eukaryotic.
Eukaryotic. Okay. Karyon means kernel or nucleus. The early microscopists thought that the nucleus of a cell looked like a little kernel. What does pro mean? It means before. And so these are cells that evolved before there was a nucleus. Does anyone know what U means as a prefix? True. Okay, they have a true nucleus, a membrane-bound nucleus. So eukaryotic cells have a membrane-bound nucleus. They have mitochondria, they have, if they're a plant, they have chloroplasts, endoplasmic reticulum, Golgi apparatus, lysosomes. Prokaryotic cells, no. Now they do have to have DNA. But the DNA is not enclosed in the membrane that separates it from the rest of the cell cytoplasm. It's another difference. Did you guys look at cells last semester at all? Did you look at cells last semester at all? Okay. When you did your mouthwash lab, did you look at the bacteria? Which are bigger? Eukaryotic cells are much bigger. Prokaryotic cells are smaller. Which one is thought to have evolved first, prokaryotes or eukaryotes? All the evidence to date indicates that prokaryotes were the first form of cellular life and that eukaryotes came later. As a matter of fact, there is an interesting and well-supported idea called the endosymbiosis theory, which suggests that both mitochondria and chloroplasts came from prokaryotic cells taking up a symbiotic relationship inside of primitive eukaryotic cells. This idea of size is really important and something we need to talk about for a second. Cell size. Is there a limit on how small a cell can be? How small it can be? Yes or no? Yeah. I mean, it's got to be big enough to at least contain this stuff, right? Can a cell be smaller than a DNA molecule? Not if the cell's got to contain DNA. Some of the smallest cells that exist are tiny bacterial cells that were at one time thought to be virus. Virus are even smaller than prokaryotic cells. But do we consider virus to be technically alive? No, we do not. There are some criteria for life that viruses don't meet. Is there an upper limit to cell size? Yes, there is. At some point in size, it becomes impossible for a cell to become too big. And the limitation that is driving that reality is the concept of surface area to volume ratio. When we're talking about a cell's surface area, we're really talking about membranes. And in the most simplistic situation, the cell or plasma membrane that surrounds the outer portion of the cell. So the more membrane there is there, the more surface area there is. Surface area becomes really an issue when you talk about cellular transport, because things are moving across that membrane. Volume basically has to do with the amount of cytoplasm that's inside the membrane. And all kinds of stuff is going on within the cytoplasm. All kinds of metabolism. All kinds of movement. And that requires energy and resources. Where do the cell's energy and resources come from? Where do they get their stuff? Yeah, from their environment. Okay. But that stuff has to be able to cross the membrane using transport mechanisms to get into the cell where it's needed. And toxins that might be produced by metabolism have to get out of the cell or the cell's going to poison itself. So this balance between surface area and volume is really critical. And if at some point in time you don't have enough surface area, enough membrane, to transport things into the cell to take care of its cytoplasm, you got a problem. So surface area to volume ratio is really critical. It's got to be favorable. Well, as the cell increases in size, its surface area increases by the square of the size increase. So if something doubles in size, if the cell's twice as big, its surface area is 2 squared or 4 times as big. But volume increases by the cube of the size increase. So if a cell doubles in size, its volume gets 8 times bigger. So what's happening here? As cells are getting bigger, there's a bigger and bigger disparity between surface area and volume. And at some point in time, there's not enough surface area to satisfy the demands of the volume of the cell. And so that limits cell size. Cells can only be so big. What's one of the reasons why a eukaryotic cell can be larger than a prokaryotic cell? How do they deal with this surface area to volume ratio problem? What do they have that prokaryotes don't have? Membrane-bound cellular structures. So in eukaryotes, they have both an external cell membrane and internal organelle membranes. And so they have a lot more surface area. Thus, even though their volume's bigger, they've got sufficient surface area to take care of that. So what we're going to do Thursday is kind of go through a little tour of some of the more prominent cellular organelles. And as I said, I'm going to try to use a white blood cell as kind of our example of a typical cell, so we can talk about these cell organelles in some kind of context. And so if you haven't been reading chapter 4, you should. And I will tell you on Thursday what the assignment will be. But there will be a quiz on Monday, a reading quiz over textbook material. For the reading quizzes, I will let you also have a page of notes. Okay? because the intent is to make you read the textbook. Okay? It'll be over chapter 4 this time, because we won't have gotten all the way through chapter 4 probably. So I'll give you the assignment on Thursday for Monday, what the quiz is doing. You can't use the page of notes on your lecture exam, okay? Okay, so we'll see you Thursday. If you have the sign-up sheet, the attendance sheet, please bring it up front up there.
Can you tell me what the quiz was? I won't be Thursday, so.
Oh, I've got to look right now. I'll send you. Would you send me an email to remind me, and I'll send it back to you?
Okay.
That's right. You and
Sam.
Sam will be gone. Thank you.
Yep. I also need to take that quiz.
Oh, right.
Do you want me to come in before my lab today?
Sure, that would work.
I have lab at like, I think, 40.
Yeah, that'll work. Yeah.
How long does it take? Like
10 minutes.
10 minutes.
Yeah.
Be here
What
by,
I,
like,
what I will
4:30.
do is I will give you, since I've given back the original quiz, I will give you back the, I will give you the first makeup one, but you'll still have, you know, if you get,
Yeah, okay.
you'll still get the same points
Yeah,
you
that's
would have
fine.
taken last time.
All right, thank you.
What do you want me to do about, because I'm missing Thursday and Friday.
You're Sam, right?
Yes, sir.
Okay.
What do you want me to do about the Thursday, Friday assignment?
I will have the